All right, it's my pleasure to introduce Jill Hudnall. She's a research associate in Starkville, Mississippi for the Global Center for Aquatic Health and Food Security, serving the Gulf Coast Aquatic Health Program. She has a BS in Animal and Dairy Science with a minor in International Agriculture and is pursuing her master's degree under the direction of Dr. Graham Rosser and Dr. Beth Peterman. Looking forward to hearing about your work, Jill.
So, hi everyone. Like you said, my name is Jill Hudnall, and today I'm going to be talking to you about the morphological and characterizing of a trematode in bottlenose dolphins. So, even though this is a fish health section, why is this important? Um, Obviously, the fish are being parasitized by these parasites, and so, of course, that's always important to know about your ecosystem. So, like Dr. Peterman said, I work with the Marine Mammal and Sea Turtle Program, where we have a partnership with the Institute for Marine Mammal Studies, where we respond to dead, sick, or injured marine mammals and sea turtles. So, a little bit of overview about bottlenose dolphins. They are considered a keystone species and a marine health indicator for water quality and pollutants. Water quality include freshwater incursions, which when there's an increased flow of fresh water, which decreases the salinity, oil spills, and of course their prey. If there are no fish, your dolphins, if there's less prey, your dolphins are either going to move or you're going to see dolphins who are not looking well. Pollutant-wise, persistent organic pollutants or POPs and polychlorinated biphenyls or PCBs are some of those. And then unusual mortality events or UMEs are a significant die-off of any marine mammal population that have to have a response. So our last UME was in 2019, and that was due to an increase of fresh water into the sound, which caused freshwater disease. This is important just because if you have lower immunity, you're going to have more parasitism, and then of course human interaction. So a little bit about the Mississippi Sound. It's 2,100 kilometers squared and includes 6 barrier islands, which include Ship Island, Cat Island, and Horn Island. And there are large passes between all of these barrier islands that allow barges or any other ships to go in and out. So there is access to the Gulf of Mexico. The Mississippi Sound has the largest stock of bottlenose dolphins in any bay or estuary with over 2,500 dolphins present. And the salinity, of course, ranges over a good variety from 0 to 33. And then we have coral reefs and seagrass beds, which is going to be important later on. So diet composition, this is going to also be important. That includes soniferous species, which utilize noise, which is important because dolphins, of course, use echolocation. And then vegetation consumption species, which include pinfish, pigfish, and striped mullid. And then, of course, your game fish like trout, catfish, and perch. So a little bit about my trematode, which is Bronina species. Bronina are classified as trematodes and involved in the order Diplostomata. There's only one genus currently within this family, which is Broninidae, and currently there's only one species, Bronina cordiformis. So definitive host, of course, bottlenose dolphins or odontocetes, which include toothed whales. And then location in the host is pyloric stomach and duodenal ampulla. We find them more often in the duodenal ampulla versus the pyloric stomach on necropsy, but they can be found in both places. So, life cycle hypothesis, of course, all we know right now is their direct host, which is the dolphin, of course. And then we're thinking, following the trend that many trematodes life cycles follow, we're thinking an aquatic gastropod is involved that the marine fish eat, which are then consumed by the dolphins. But we're not sure if metacicaria is encysted inside the vegetation that the fish are eating, and then involuntary eating the snails, and then the predator fish are just eating the snails and the grass is just involved. That part we're not sure about. So this is just an overview of the life cycle. This is all we know, of course, is the dolphin. That's all we got, all we know about this. And then we are thinking eggs are passed into the feces that either insist in the snail and or vegetation. We're just not sure yet. And then the fish consume either or, which are of course ingested by the dolphin. So a little bit about the vegetation within the Mississippi Sound. This photo is a study completed in 1992, and this is a photo from 1999 of the seagrass that's located within the Mississippi Sound. It surrounds most of the islands located, and then there are some mainland locations. So this top photo is shoalgrass, which is the most dominant, and then turtle grass, and then widgeon grass. I think it is important to note, after I've just been looking at the different varieties, widgeon grass is grass, but it's not grass because it doesn't really stay within the ground. It just kind of floats along collecting debris. So a little bit more about Bronina. Original descriptions include Carl Hyder in 1877. So this is a really old worm. He really only described the external description, and he assigned the genus Bronina. And then Carl Wolf in 1903 used the same specimens and just piggybacked off a Hyder, and he described the internal reproduction organs and then organs just in general. However, he was credited for labeling a species, Bronina cordiformis. And then Scraven in 1963, he continued that description, and he spoke about the eggs, which nobody else had done yet, and then the attachment to the host. So host attachment, they are attached by a stalk-like spicule seen here, of course, and then everything that we see most of the time when it's drawn, you see that the stomach mucosa kind of goes up into the mantle that the Bronina has. However, when we look at histology or when we stain them, we don't see that. So this is just my personal hypothesis, is that as the parasite matures, this recedes back, and then this just becomes empty. So I feel like the immature trematodes have to do this, and then they become encysted. So morphology, so the adults are encysted within the mucosa. They are heart-shaped, which is where cordiformis come from. Cordiformis means heart. They have a stalk-like spicule, and they have a serous spicule, which you can see there. So the eggs are operculated and amber-colored, and they're very large. Dr. Rosser likes to call them the big boys. And then pathology, they don't really do a whole lot, to be honest with you. They may cause some pinpoint gastritis. There was a case study I found where a dolphin had over 900, so maybe an impaction process, but we have never seen that. So distribution, the first description was in the common dolphin in the Adriatic Sea by Hyder in 1877. And then they really have just focused on the southern hemisphere. We have a report in South Africa of a bottlenose dolphin. And then we have several reports in South America. So Chile, Brazil again, there's a lot of Brazil, just Patagonia in general. The most southern tip of Argentina islands, Tierra del Fuego. And then several more reports in Italy again. So the latest report we have is an Indo-Pacific finless porpoise in Thailand in 2018, and that was the first report that we've ever had in Asian water. So this is just an overmap. Here, Italy, this is your Adriatic Sea. That was your first report of Bronina. And then the last report, or the last novel finding, was here at the southern tip of Thailand. So these specimens, this is what you see on necropsy. We have specimens that we've harvested from 2019, from 2024. We also only have 10 specimens, so they're few and far, they're common, but they are few and far between. necropsy, it varies across ages. We don't only see these in adults, and we don't only see these in immature animals. We have specimens from calf all the way up to adult. And then we store them in ethanol, 10% formalin, and then we freeze them back for just if we need some more. So molecular data that we have, GenBank has a few entries. Specifically, they have Bronina corniformis categorized there, but we use DNA extracted from an adult trematode to do next generation sequencing for OX4, nanopore, and Illumina to create that genome. And whenever we sent that off, we got it read back. And there's 2 different base pairs in comparison for our trematode and the trematode Bronina corniformis. That means we probably have a new worm, and I don't know how I feel about that. So really, there's a 91% difference between the sample we submitted versus the sample that's already labeled Bronina corniformis. So our future plans include examining fish and aquatic vegetation and the snails to really determine that life cycle because nobody knows it. Everybody just says Bronina corniformis, it exists, that's it. And we would really like to start that off with evaluating the stomach contents because we're just not sure if this specific family of fish or just general consensus, just fish in general. And then, of course, morphologically characterizing of a potential new species. By doing that, we're going to do histology. I did turn in our first trematode into histo last week for that to be trimmed in so we can evaluate it. And then just looking at and excising different organs. So acknowledgements, of course, the Global Center for allowing me to work full-time and get a master's degree all at the same time. And then Dr. Peterman and Dr. Rosser for being my co-major professors and who I ask a lot of questions. And then, of course, the necropsy and stranding staff. These are all my references. And are there any questions?
Great presentation, Jill. Very interesting
Thank you.
project. And I think it's okay to be excited about finding a new.
I told Dr. Rosser when he told me that, I was like, this is getting more complicated than I thought. I don't know how I feel.
Folks, if you have questions, feel free to unmute yourself or you can drop them into the chat. Okay, I've got a question for you, Jill. I'm curious how much time you spend doing surveillance of the dolphins versus waiting for them to die and wash up somewhere to be recovered.
So for the Global Center, we only work with deceased dolphins. But basically every dolphin that strands on the Mississippi Sound, like Dr. Peterman mentioned, that are early coats, which is just a level of decomposition, we're going to necropsy those. So our surveillance really just includes any bottlenose dolphin that's died within the Mississippi Sound that was able to be recovered. We're looking, we do a complete necropsy on those. And so we always evaluate stomach and stomach contents and any additional parasites like that. Live, I don't think we'd ever be allowed to look at live dolphins, but I think the fish is going to play a big part in that. It's just going out. That's going to be our plan for this summer is to go out via a charter boat or somebody else to collect these fish. That way we can bring them back and evaluate them.
Awesome. All right, well, thank you very much for your presentation.
No, thank you.